Thanks for giving me the opportunity to introduce my research here. So today I would like to talk the research about how we harness the power of machine learning and omics to identify the microbial function in composition across diverse environmental conditions so that we can use this in the Earth system prediction. So I'm Yang Song. I'm a assistant professor from University of Arizona. So first, I always want to start this talk from this very big background. We know that the current Earth system model still had very large uncertainty when they predict and project the future climate. So if we see the figure here on the left that's showing from the different Earth system model, if we look at the projection for the future global mean temperature, the very uncertainty here can be 2 degrees. So we know that the pair agreement try to mitigate the future climate warming by 1.5 degrees. So compared with that magnitude, we know this uncertainty is very large. Then when we try to see where those uncertainty come from, we see that compared with the uncertainty coming from the interaction between the land and between the atmosphere and the ocean, we see most of this uncertainty actually is coming from the land and atmosphere, like the figure in the middle here showing you see this very large uncertainty coming from this flux estimation. Then we will further check that where this uncertainty about the carbon flux comes from. So one big uncertainty here actually coming from the climate and soil carbon feedback. This is a very typical and classic Earth system model research, which first try to utilize and analyze when we consider the microbial function in the Earth system model, how that may affect on the future projection of soil carbon. So here we could see that here from the for the future projection, we either use this DaySand and CM14, that is the Anka community land model, and both of these models, they have not really explicitly represent the microbial function. So they only consider that the soil physical process will affect on the decomposition of organic matter. Then another, these two models, the CM Max without the adaptation to the warming. So basically that they already use the materialist mental equation to represent the microbial in the Earth system model, but they have not consider whether it will affect when the climate change, whether it will affect on the carbon use efficiency or any kind of the microbial feedback. Then this line basically that showing that is considered under the future climate condition, microbial community basically can adapt to it so that it will adjust its behavior to mitigate that kind of the uncertainty. So basically, we can see that this different model really showing very diverse projection for the future climate condition. So this gives us the idea that one important issue, if we want to mitigate the future prediction of carbon climate feedback, we need to better constrain the microbial function in the Earth system model. But currently, we have several challenges about this. So first, that relate to the diversity question. Because of the microbial community that are very difficult to be observed, so we don't know how to diverse microbial community will regulate the composition of soil enzymes and also their ecosystem function. Another thing that relate to the dynamic question, basically how the microbial community will respond to the climate change, the environmental change, and how this feedback will further affect on the biogeochemical feedback. This is also a question. Another question here that relate to the heterogeneity. So because very diverse environmental condition, so that also will introduce very diverse distribution of soil enzyme composition. So without answer these three questions, basically we still have a very big challenge to just incorporate the microbial function into the Earth system model. But the good news is that I think that is more related to today's our effort here. So omics technology development and also the mass spectrometry technology that allow us to further discover what happened in the below ground. So basically, we know that from the omics data, we can use that to identify the diversity, dynamic, and even heterogeneity of the soil microbial community. But the question here is that when incorporate those information to the larger scale, for example, to the soil biogeochemical cycle, there's still a scaling issue we need to overcome. So I usually summarize this kind of the challenge as these three components. The first that is data complexity and also the functional redundancy. So we know that the omics data is very complicated. Another thing is that the omics data, even for the same gene, they coming from different microbial community, they may play the same role, just decompose a very similar chemical compound. So all those introduce basically the complexity for us to utilize this information into the biogeochemical process. Another that most of the omic data, they are the nucleus data, means that they usually indicate only the abundance without have the mass unit, so that it's difficult to link that with the most of the model which try to trace the mass chains of the organic matter. And another thing is that because the omics data that usually coming from the soil sample is very small scale. So how we upscaling this small scale information to the biochemical process that still is, especially in the Earth system model, both the temporary scale and also the spatial scale is larger compared with the soil sample we get. So how we can use all those information. So basically since the 2016, I have worked on this field to try to overcome all of those challenges. So when we already developed an approach to help us to overcome the challenge related to this data, omics data redundancy, and also how to upscaling this to the biogeochemical model. Our first effort that we use the soil sample we collected from the tropical site that in Panama, that basically a location where it has very diverse soil microbial community. We use the omics data collected from that soil sample. We classify, we first extracted around 2,000 enzymes that relate to the basically the soil decomposition, carbon, nitrogen, phosphorus decomposition process. Then, in order to further simplify this data, this 2,000, that is all the data related to all the soil process. Then we only extracted this 108 enzymes that only relate to the carbon, nitrogen, phosphorus decomposition, and also the nutrient mineralization. So then, based on this information, we further group it as this different enzyme functional group. So we identify a concept that called the enzyme functional class. Each class that is a group of the enzyme, they basically catalyze a very similar chemical compound. For example, this is a group of the enzyme that is related to the carbohydrate decomposition. And then this is a group of the enzyme that relate to the lignin decomposition. We also group a group of the enzymes that relate to how to catalyze a very large protein compound to the smaller amino acid. So through this approach, basically we can simplify the complexity of the omics data. And here we also use the mass spectrometer information to help us better identify this pathway and whether those enzymes really play the role in those different pathways. So that's basically our starting point. Using this information, we already developed a set-level biogeochemical model, and we realized that when we use this information to represent the microbial function diversity, that help to mitigate the future soil carbon emission and the global warming condition. But this conclusion here, we are still have to questionable it because that this is just a one set level. So basic question here for us try to utilize this approach into a more larger scale. We try to answer these three questions first. Does those general enzyme function class as we found in that one site can evolve in the soil, carbon, nitrogen, and phosphorus cycle can be found in other very diverse environment conditions? And also how does this enzyme function class composition will vary across the diverse environment? And can we use the machine learning model to do the prediction for the EFC composition so that because of course, the omics data sampling is still very limited. It's not covered a very diverse domain. So if we can use machine learning to predict their enzyme functional class, so that will help us to further utilize this information into the larger scale. So today, I will mainly introduce how we answer these three questions. To answer these three questions, we actually develop this whole machine learning pipeline. So our machine learning pipeline starts from we collect those existing published omics data. Here is mainly use the basically the metagenomics data. Then we collect those from JGI and also the NMDC. We also pair those data with basically the corresponding environmental data. So here we collected those around basically 300 this metagenomic sequence data from the around 20 sites across the whole US, including the forest, the grassland, the different kinds of the land cover, and we paired those with the around 19 biotic and also a biotic environmental condition, for example, the soil texture, soil chemical, and also the climate condition, even the above ground vegetation, like their the productivity of the vegetation, all those information. So we use this as our basically the training and evaluation data. We use this paired omics information with the paired environmental information. We train our this example machine learning pipeline. So in our machine learning pipeline, this machine learning has two properties. The first that we introduce a data augmentation approach. So basically, because only around this 300 omics paired data is still very limited to perform a better machine learning analysis. So in order to improve the prediction ability of this machine learning pipeline, we follow the idea from the machine learning field. They usually use the augmentation to analyze the different image first, so they can change the shape and change the the angle of those things in the image. Then they use this way to augment the model data. Similar things here that we can see that if we randomly pick out one enzyme that accounts for very small relative abundance from the whole database, so that we can build a augmented model database compared with our original data sample. So another thing here, we use this Monte Carlo cross-validation approach-based multi-example pipeline. So we introduce a different machine learning approach here, for example, the random forest and also the bagging and the boosting. We try to utilize the prediction power from this different machine learning approach. Then finally, we assemble the prediction of all those models together so that we can increase the prediction ability of this whole process. So basically, this process finally, we will also use testing data that we collect the environmental data from around 1,000 soil sites across the US. And this includes all the environmental factors we consider here. Then we use that to predict what could be the spatial distribution of the enzyme functional class across the whole US domain. So here I will show you our first result. First, we found that we can answer our first question, enzyme functional class can be predicted using the machine learning model, and we developed here. The figure I'm showing here, that is the one matrix, that is R-square, to evaluate the model ability to predict the different enzyme functional class. Here from the left to the right, that indicates the this EFCC, that is means all the enzymes group. We classified they relate to the carbon lignite decomposition. This EFCN, that is all the enzyme related to the nitrogen-rich organic matter decomposition and mineralization. So again, this one EFCP, that is related to the phosphorus-rich organic matter decomposition, all those enzymes. And similarly, if we see from the left and to the right, you can identify this by the name, that is all the enzymes related to the carbohydrate, lignite decomposition, organic nitrogen monomer nitrogen, inorganic nitrogen assimilation. So the same thing here, that's for the phosphorus. Then the last column here, that is the overall performance for all the EFC we classify here. Then the blue bar here, that's basically the model training data result. Then the yellow bar that indicated the result for the evaluation data. So basically, we can conclude here that the model did a good job to predict the enzyme abundance for each of these enzyme function class. But compared with those predictions for the carbon and the nitrogen related to the organic matter, basically we see that the one related to the phosphorus will has a less prediction ability compared with them. Actually, this is related to in our environmental data which we collected. We are still lack of few data related to the phosphorus. So, for example, like the organic phosphorus component. So those actually affect on this research result. Then in another way here, we also use a telegram to showing how the model's performance. Here, just one example for all the enzymes related to the lignin cellulose decomposition. So we can see here that all the different sun here, the color, this is related to the different ensemble model we utilize in this machine learning pipeline. Then this is the final, the broad color circle that indicate the final ensemble analysis. So if we read this figure, we can see all their correlation is around 0.99. So it actually means all the model do a very good job. They get a convert prediction. Another information we can learn from this figure, basically this red star here indicated the standard deviation of the observation data. So it means that the omics data we used to train the machine learning model. Then if we compare this value here showing this, so basically if we look at the each of this value that's indicated if this all those stored more close to this one. So this indicated basically the prediction error RMSE is among this value. It's almost 30% of the total basically the total standard deviation of the observed omic data, because this is around 6. So this also gave us idea that basically the current model's prediction error is acceptable. Another information that we see this the standard deviation of the prediction is also very close to the standard deviation of the omics data. So that indicates not only capture the overall information, our model can also capture the basically the spatial variability of the EFC abundance related to the lignin decomposition across the US domain. The same thing here, we also check those EFCs for the nitrogen-rich and also the phosphorus-rich organic matter decomposition. We found the very similar result, but again, for the phosphorus compared with the nitrogen and the carbon, their prediction ability is relatively low compared with another two. So this is the our machine learning result. Then after using this machine learning, we incorporate all those around 1,000 environmental data into this machine learning result. We predict how the enzyme function class related to the soil carbon, nitrogen, and phosphorus cycle will be distributed across the US domain. Here again, to be simplified, I only show that those enzymes relate to the lignin cellulose, those enzymes relate to the nitrogen-rich organic matter, and enzyme related to the phosphorus rich organic matter decomposition, how those different group of the enzyme function class distributed across the US domain. So basically, we can see that those general enzyme function class, which we identified from this 180 enzymes that group that into this around 8 enzyme function class, they can be identified across very diverse environmental conditions. However, if we look at their relative abundance, that can be very different even when we go to the different location. Basically, we see the relative enzyme abundance for those for decompose the carbohydrate and also the lignin is relatively larger compared with those for the nitrogen-rich organic matter decomposition and also the phosphorus organic matter decomposition. That is showing some domain difference. So later, we try to use our machine learning tool to better understand what environmental factor is important to introduce this diverse condition. So this is an important figure showing the importance of different environmental features we use in this machine learning model. So this is for the EFC enzyme function class for the lignin cellulose decomposition. So we can see that the very important factor to make this prediction is more related to the soil chemistry process, like the soil-syn ratio and also the soil carbon. And another that also relate to the basically the above ground vegetation information, like the land cover, what kind of the vegetation is covered here, and what will be the MDVI here, that is the remote sensing data to indicate the productivity of the vegetation. And the elevation here, all this is actually related to the temperature condition. And so that is all the important factor to introduce the basically the, oh, okay, so I think I almost finished to introduce the, I run the time, okay. To introduce the information. Then if we look at this, is also very similar for those model for the EFC, for the natural enriched organic matter deposition. And then we look at the phosphorus, because of our data is actually distributed from the surface 10 centimeters until to the 1 meter layers of the different soil sample. So we see the soil depth play a very big role here to do this prediction. Then if we further use this information to understand what's the relationship here, so we can see that it's actually show a very clear relationship. We see for the soil carbon, which is higher than 1%, basically that will support the relatively high enzyme function class related to the lignin cellulose decomposition, but relatively less enzyme function class related to the nitrogen-rich organic matter decomposition. And here that is showing how the soil scene then control the partitioning of the enzyme function class related to the nitrogen-rich and phosphorus-rich decomposition. Another intriguing thing here, I want to show that soil texture, that is a very important factor to determine where the enzyme will, the microbial community will use the nutrient from the different kind of substrate. So for example, here you can see for those very fine soil condition, basically it's totally showing that they will support the relatively less organic enzyme function related to the organic phosphorus decomposition, but a relatively high enzyme function related to the inorganic phosphorus assimilation. And when we incorporate all those enzyme function class together, we'll realize that they strongly correlated to the precipitation. Basically, in most of this relatively dry location in the US, the total enzyme function class related to this actual cellular decomposition process is relatively low compared with the others. So this indicated that basically we can use this model to interpret some life history strategy of the microbial community. Okay, so this is my take-home message here, and basically the 3 questions I mentioned today, and we thought that our machine learning model can answer that. And we are also integrating the more omics data and also the high resolution environmental data like the mass spectrometry data to help us to further mitigate into this machine learning pipeline. So if you are interested in our research, please feel free to contact me. Okay, thanks. Sorry for run of time a little bit.
Thank you, Dr. Song. I think we have time for maybe one question before the next speaker. Does anybody have any questions?
Yeah, I had a question.
Okay.
That was amazing. The 118 enzymes that you used, were those based upon a priori expectations of particular enzymes being a part of a certain degradative process, or was that something like, yeah, this is what we found, 118 enzymes? And the second part of the question is, how many different genera were those enzymes in? I mean, did you have repeated enzymes across genera A, B, C, and D, multiples, in other words, of a given kind of enzyme?
Okay. So thanks for your question. That's very important question. So first, that for those 100 enzymes, basically how we identify is that we, based on the original soil sample we got from Panama, we first use the metagenomics, and at that time we also had a metaproteomics. We use these 2 data to help us to constrain whether those gene-coded enzymes existed in the soil. And then we also have the field sample to do the mass spectrometer, actually use the EMSL technology. So at least we can see that those lignin or cellulose, those chemical compounds show some abundant difference. Then we use all those information try to help us to have more confidence that at least those pathways really existed, and at least have more probability to exist. So basically that is our way to identify this 118 enzymes. Yeah.
Thank you.
Thank you.